All right, while we're waiting for the others to come in, we'd like to officially begin session 3, which is stem cells. And I'd like to introduce our moderator for this session from the Department of Animal Science and Biotechnology of Chung-Hsing University here in Taiwan. Let's all give a warm round of applause to Dr. Cheng.
Thanks a lot. And the next thing is we got two speakers. First speaker, we invite Dr. Seiki Haraguchi. Yeah. Haraguchi got his PhD degree in 1998 from the Department of Reproductive and the Development of Biology, the Institute of Medical Science, University of Tokyo. And he did his research on 2010 until present as a senior researcher at the Animal Breeding and Reproductive Division NARO Institute of Livestock and Grassland Science. Other than 2006 to 2010, is the chief at the Laboratory for Embryonic and Genetic Engineering and chief at the Center Research Institute. And 2003 to 2006, he did the research associate at the Western Trust and Cancer Research at UK Wharton Institute, University of Cambridge. And today, he will give us the talk. The establishment of the NIAS is cell, like cell in pigs. Dr. Seiki Haraguchi, please.
Thank you for the introduction, Dr. Chen. So I'd like to appreciate all persons concerned giving this kind of opportunity. And I also would express my thanks to Drs. Wu and Tsai taking us to the downtown last night for nice food and sightseeing. So today, what I'm going to talk about is establishment of embryonic stem cell-like cell in pigs. Embryonic stem cells are derived from early stage of embryos, mainly from inner cell mass of blastocysts. ES cells have two distinct properties, self-renewal and pluripotency. Under defined condition, ES cells are capable of propagating themselves indefinitely. And ES cells also pluripotent, meaning they are differentiated into all derivatives of 3-germ layer, endoderm, ectoderm, and mesoderm, including germ cells. To date, the ES cell contributing germ cell have been reported only mice and rat. The first report of ES cell is in mice in 1981, followed by pig, human, and rat. Although rat is classified as rodent, it took 27 more years for the establishing of the ES cells, which is one of the great milestones in the field of stem cell research. The Austin Smith group found the importance of signaling cascade involving self-renewal and pluripotency, such as ARC and Wnt signaling pathway. And blockage of this signaling using specific inhibitors enabled to isolate rat ES cells. So until recently, many researchers tried to establish true porcine ES cells, as yet not yet succeeded. So the reason why the establishment of true porcine ES cell is difficult is it is said that the general method for the establishment of mouse and human ES cells are not applicable to the case of pig, and the cell cannot maintain for longer passage. There is no reliable marker for the evaluation, and porcine ES cell cannot form embryo body. This table shows characteristics of mouse and human ES cells from the features. Generally, ES cells are classified into two groups. That is mouse type and human type, or naive and primed. It is said that most typical differences between them are colony shape and factor requirement and the status of X chromosome activation. So you can see the dome-shaped colony in mouse ES cells. And this is a human ES cell, like a flat colony. If is leukomia inhibitory factor, which is necessary for maintenance of the pluripotency and cell renewal for activating STAT3 in mouse ES cell. On the other hand, basic FGF is indispensable for human ES cell culture. Upon the process of establishing porcine ES cell, we refer to the feature of mouse ES cells. Particularly, we pay attention to the colony shape, if-dependent growth, status of X chromosome, and effect of inhibitor. But as you may suppose, it is not necessary to say that putative established porcine ES cells are in the naive state if the morphology is agreement with those of mouse ES cells. After all, they need to be confirmed by their contribution to germline chimera. This is outline of establishment of porcine ES cell-like cell in our laboratory. Inner cell was mechanically isolated and put onto the feeder cell, which is mitomycin-treated mouse embryonic fibroblast cells. So we use initially the FCS-based ES medium in the initial stage. So this is a primary colony derived from ICM, inner cell mass. Then we change to the KSR-based ES medium to minimize and to replace the differentiation. We define as established cell line when they were satisfactorily grown after freeze and thawing. Here you can see that the morphology changed before and after addition of inhibitors. Firstly, the colony shape changed to the compacted like a dome shape with high cell density, and doubling time was shortened from 5 days to 2 days passage culture. And the colony could be maintained without cellular senescence for longer passage. To date, we established several cell lines. Even though not all cell lines can be maintained satisfactorily, some of them are differentiated at some point, which means our cultural condition is not consistent, not perfect, and there is room for the improvement. So I explained about established cell line in detail. These figures show undifferentiated markers protein expression established cell line. They express undifferentiated marker SSEA1, SSEA4, OCT4, and NARO. In mouse ES cells, SSEA1 is positive, and human ES cells, SSE4 is positive. But in non-established cell line, both are positive. The expression of OCT4, which is best known as undifferentiated marker and key factor for ES cells, are relatively lower than that of mouse ES cells. Also, we wanted to study as a major undifferentiated marker protein, the cross-reactivity of the antibody is always bottleneck. So as I mentioned, there is no reliable marker in the porcine ES cells. It is not known which gene and how much amount of the expression level is important. But anyway, we comprehensively studied mRNA expression, the general undifferentiated and differentiated markers. So I'm just letting you know that this part of picture is cut and pasted for this presentation, but it doesn't affect for the application. The expression level of gene is different among the individual cell line, suggesting that clonal variation. And most of genes expression level are lower than those of blastocyst, OCT4, NARO, SOX2, and so on. Contrary to our expectation, the mRNA expression patterns were not dramatically changed before and after addition of inhibitor. Although the small upregulation and downregulation was observed, we can't explain the reason at this moment. This slide shows the telomerase activity and X chromosome activities. The established cell line have high level of telomerase activity in both early and later stage of cell line cells. This is mouse ES cells. Telomerase activity is also high. The right panel shows maturation analysis of exist gene by COBRA combined by sulfide restriction analysis. As a result, The X chromosome status of the cells show active and inactive XA, XI. So same as somatic cell and human ES cells. This slide shows that cell lines, the established cell line propagate dependently. When the leaf was deprived from the medium, they can't be maintained. The cell express leaf receptors, and it was proved that leaf activate the STAT3, suggesting that leaf STAT3 pathway is functional similar to mouse ES cells. According to the general method, we tested teratoma formation assay using the established cell line, which maintained on the feeder cell, and embryoid body-like sphere, constructed in suspension culture for several days. After 2 months of inoculation, we could confirm something small changes. But typical teratoma formation could be observed either at the microscopic and tissue level. These are hematoxin elgin staining. There seems to be muscle and cartilage in it. But if so, the further analysis is necessary by using appropriate antibody. So we attempted to establish the stable cell line expressing GFP and RAGZ marker genes, and successfully obtained them after G418 drug selection. Using this cell line, we asked whether the established cell line have ability to contribute to the interspecific chimera, because peak chimera analysis is not always available in many facilities as well as in our facility. It seemed that the cells were incorporated into embryo, both mouse and chick. We are currently studying the chimerism at the later developmental stage, and to see whether interspecific chimera, this analysis useful for evaluation of pluripotency of established porcine ES-like cells. This is a summary and comparison between mouse and human ES cells. The features of established porcine ES-like cells are partly consistent with those of mouse ES cells. The dome-shaped colony, leaf-dependent growth, and self-renewing response to inhibitors. But still not clear about pluripotency. The X chromosome activation in both alleles may be important, as previously reported. We have been studying on establishing true porcine ES cell. The reason is very simple. We want to know whether it is possible or not. So what to do next? So finally, I'd like to introduce our ongoing work. As I mentioned, the expression level of undifferentiated marker genes, such as OCT4, is relatively lower than comparing blastocysts and mouse ESS. So we are attempting to introduce such molecule to the cell derived from ICM, inner cell mass. So at this moment, we could get many cell lines efficiently, and we will see what's happening in the near future. So these are our collaborators. Dr. Nagai was our previous boss. Thank you for your attention.
Okay, now we open for discussion. If there are any questions. No? No, please. Yeah, I got a question now. I try to. The usual use protein, particularly for those very early stage structural cell, those is gene, and try to control and use the strength and try to use them as a feeder layer. Those options transform the hypoproximity cell and try to maintain them. Use this demonstration to try to maintain the unfold image. We'll give this part. Try to from Indonesia,
Yeah.
such as for we got stuff like this, you from there,
Yes.
then try to control it.
Yes.
Use for then you use for time or transfect the cells.
Yes.
After the layer, try to maintain their multi-spirit differentiating potential. Okay, very much better.
I introduced such kind of genes by RNA and keep maintaining such cell on the feeder layer, and it's going to the passage culture at least more than 20 at this moment, but it's possible for longer passage, but I didn't yet check. This is ongoing work.
How do you maintain try to use those kind of cell line for detect the visual differentiation potential? That's the problem. Actually, in fact, we also did some of this kind of study. Use the pig, use the mouse as a model. Of course, from initial we use the mouse as a model, and eventually get into the pig. I mean, those are very early stage. You try to maintain this kind of stem cell for multiple lineage potential. Yes, right. Suppose they should cut potential to expression of early gene for maintaining multiple expression potential. And you introduce this kind of gene as kind of useful to model structure.
I didn't use a vector system, just introduce RNA directly. So as long as I keep doing the transfection, they maintain their morphology like this shape.
Like this differential potential, differentiation potential.
I'm checking from now. I don't know yet.
Yes, that's a Any more questions? Thanks a lot.